Oops, sorry. I better go to the... All right, are there any questions from last time? Okay, the exam will cover until the end of transcription and translation, okay? So everything is fair game, and I thought I should let you know today because that way you can start studying, it's still 1 week away, okay? So far maybe you've been relaxing, but now is the time to, start firing on all cylinders. I don't know right now, yeah. There'll be descriptive, see, time is not going to be a factor, okay? Like if you're not able to do something, it will not be because there are too many problems, okay? It'll be because you didn't study, okay? So you have to start studying, right? Right? And it's different from like doing 314 or 214 or whatever, right? Because there's quite a bit of memorizing and, but that's the power of repetitive learning, So some work you have to do, That is the nature of biology, okay? We cannot boil it down into some equations or something like that, so. But yeah, this exam will be like that, the next exam is like that, But then after that, it's going to be engineering stuff, so. Okay. But you need to know the biology, and this is your opportunity to learn it, because if you don't learn it now, you'll never learn it. Yeah, go ahead.
You said the coverage is?
Up to the end of transcription and translation. Okay, so that means probably is the first, is it six chapters, I think, yeah, because chapter 5 is DNA, there are 4 is protein, and I think transcription and translation is chapter 6, yeah. Okay. So it's not too bad, and purpose, you're getting more time because now I'm doing genetic variation, so you have more time to read that stuff because I covered it, maybe 2-3 lectures back. And even for the second exam also, you will get a lot of time. It will not be that I covered stuff today and then you have to come and take the test on Monday, You probably will get 1 week to 10 days. And we'll be doing other things, mathematical stuff, but you will get time to get familiar with the biology. Okay. All right, this is the chapter on genetic variation, right? And in this chapter, we will basically look at, changes in the DNA that occur over evolutionary time, right? So although cells go to great lengths to maintain the integrity of the DNA, permanent changes in the DNA, which are called mutations, do accumulate over time, right? In fact, the vast diversity of life that we see around us today has arisen through changes in the DNA that have accumulated over evolutionary time. And this genetic variation or the diversity, it refers to the genomic differences between different species as well as between members of the same species. Right? Even members of the same species don't have exactly the same DNA. Right? In fact, no two human beings have got identical genomes, that means identical DNA sequences, unless they happen to be identical twins. Right? Now, it is generally believed that the conditions on this earth have undergone dramatic changes over billions of years. Right? So in order for life to propagate, it is essential that the survivors have to be able to adapt to the changing conditions. Like if the temperature on this earth becomes like 200 degrees or something like that, most of us will be dead, okay? But there'll be a few of us probably that will survive, right? And then for us it's no big deal. Like there are bacteria that can live in the vents of volcanoes, okay? They're called thermophilic, heat-loving bacteria. They can live there, okay? So it is believed that genetic variation, although not always beneficial, is responsible for conferring survivability in a changing environment. And there are 3 main mechanisms by which genetic variation can arise in nature. The first one would be rare mistakes in DNA replication and repair. Right? Every time you're copying DNA, there's a small chance of making a mistake. Okay? These could accumulate over time. The second one would be the recombination of DNA and the activity of viruses and other mobile genetic elements. Right? So in this chapter, we'll talk about what viruses are, right? That can move into and out of the DNA and make changes. And the third one that causes a lot of genetic variation is the reassortment of the gene pool of the species into new combinations during sexual reproduction, right? Because in sexual reproduction, you inherit traits both from your mother and your father, and there are lots of different combinations that you can have. Now, first we'll look at genetic variation in bacteria, then we'll look at genetic variation in eukaryotic cells. Now, among bacteria, E. coli is a model organism for genetic studies. E. coli is said to have a haploid genome, right? Because it has only 1 copy of each gene in its genome. We have 2 copies of each gene, right, in our genomes. E. coli has only one copy. So it's easier to study because in E. coli, if you go and mess up one of the copies, right, you will see the stuff at the observational level right away. Okay. Whereas in the human cell, if you mess up one copy, the other copy might still carry you through. So you may not notice the effect. Right. So we are diploid organisms means we have two copies of each gene. Right. So a DNA for humans, a DNA mutation in one chromosome will not necessarily result in an observable phenotype. Right. Okay, genotype and phenotype, you remember the terminology from before. So in this context, let's recall how an individual with only one defective beta globin gene is not going to exhibit the full-blown symptoms of sickle cell anemia because that person has the other gene, just 1 minute, has that other gene that can still produce normal hemoglobin. Okay, they will feel tired. I used to have a grad student that was a carrier of sickle cell anemia, and he told me he used to feel tired quite often. Yeah, go ahead.
I have two copies of each gene in diploid.
Organisms, yeah.
Totally the same.
What's he asking? Yeah, I didn't get it.
He means if the two copy of each gene is totally the same, right?
No, they will not be exactly the same. Okay? Because like you have, let's say, two copies of each gene, right? One you got from your mother, one from your father. So it'll be a little bit different, right? A little bit different. No two people have exactly the same DNA. So it'll be a little bit different. But functionally they'll be the same, okay? Because since you're here attending my class, I assume both your genes are functioning, right? Because if they didn't, you wouldn't be here, right? Or I wouldn't be lecturing here. So functionally they are the same, but they're a little bit different. Right? It'll not be exactly the same DNA sequence, yeah.
Only the identical twins.
Yeah, identical twins have exactly the same genome, right? Because they are from the same fertilized egg that has been split. Okay, so yeah.
So why siblings cannot have the same?
DNA. Let me get to the end of this chapter. It'll become crystal clear to you why you won't have the same DNA. Because the odds are it's really huge. Your chances of getting hit by lightning are a lot higher than that. Because the odds, the combinations. Okay. Because see, I'll point that out when I get towards the end of this chapter. But look, there are 23 pairs of chromosomes, right? You inherit 23 chromosomes from your father, 23 from your mother, okay? Right? So now each person can produce, even if you don't look at other possibilities, they can produce two different kinds of gametes. Because even your father, okay, or your mother, right, these two chromosomes, they'll give you one of them. But they also got two of them from their parents, right? So for each chromosome, there is for each egg or sperm, there are two possibilities. On each chromosome, there are two possibilities. If you look at across 23 chromosomes, it's 2 to the power of 23 possibilities. Okay, that fertilized by, like an egg fertilized by a sperm, 2 to the power of 23 times 2 to the power of 23. Okay, so what are the chances that two people will have the same genome? So you won't. But anyway, I'll do some calculation towards the end of this chapter to convince you that that's not going to happen. Right? Now, E. coli, like other prokaryotes or bacteria, it reproduces by a fission-type cell division. See, eukaryotes, they do not, they have what is called mitosis because inside a eukaryotic cell, you have the nucleus. Okay, so there has to be a nuclear division. Okay, and that nuclear division is called mitosis. In the case of eukaryotes, there's no nucleus. There's just cytoplasm inside, and there's a cell membrane. Okay, so the DNA just replicates, and usually it's a circular DNA molecule, right? And then the two replicated DNA will move towards the two ends of the dividing cell, and the stuff will pinch in the middle, and you have two bacteria, right? So the DNA replicates, the two identical strands move to the two ends of the growing bacterium. The bacterium then splits into two, producing two daughter cells, each containing a genome that is identical to that of the parent, right? Now, in the presence of sufficient nutrients, a population of E. coli doubles in number every 20 to 25 minutes. Okay, so if you do the math, in 24 hours, you'll have billions of them. Okay, so in a single day, in less than a day, an E. coli can produce more than 5 billion descendants. So you have more bacteria than you have humans on this earth. Okay, in 2 days, so you're going to lose the battle. Okay. Now, every time that a cell divides, the DNA has to be replicated, which means that there is a possibility of some mistakes. Right. Now, the rapid rate of division of E. coli means that very large populations of E. coli cells in which DNA mutations have occurred can be produced quite rapidly. Okay, you hear about bacteria developing resistance to antibiotics and all that. because it will develop the mutation, okay, and so it can survive the antibiotic. And this can be illustrated using an example. Now, E. coli bacteria can be quite easily used to demonstrate how DNA mutations will confer survivability in a changing environment. For example, consider the antibiotic rifampicin. Okay, I have never been prescribed by this one. I haven't, like you hear about, amoxicillin, and others, but I suppose this is also one of the antibiotics. Right? I don't know why they picked it. But like many other antibiotics, the way they work is that they will bind tightly to RNA polymerase, bacterial RNA polymerase, inside the E. coli cell and prevent it from transcribing DNA to RNA, right? Now, if the cell cannot, transcribe DNA to RNA, it cannot produce the proteins that it needs, and the cell will die, right? The E. coli bacterium dies. However, in a large population of E. coli, say you have a billion cells of E. coli, there will be some cells which are rifampicin resistant, okay? So if you treat a population like this with rifampicin, most of the cells will die, okay? But the few that survive, right, they will keep on dividing, and remember, in a day you're going to have 5 billion, right? So they will take over the population, right? And this rifampicin resistance here comes from mutations in the DNA, which will allow RNA polymerase for those those mutant cells to transcribe even in the presence of rifampicin, right? So the treatment of using antibiotics is based on that, going and blocking RNA polymerase, right? Bacterial RNA polymerase, right? So that transcription cannot take place. All right, any questions? So at least you learn now how antibiotics work, all right? Learn something, all right? Now the principle that we have encountered here, namely the ability of antibiotics to block bacterial transcription, is the basis for current day treatment of bacterial infections using antibiotics. The reason that this treatment is ineffective against viral infections, because if you have a cold or something, right, they don't give you antibiotics, right? It has to heal by itself, right? I mean, is that a virus doesn't have its own transcription and translation machinery, which the antibiotic could have targeted. So if somebody gives you an antibiotic to kill the virus, they will kill you because your cells will not be able to produce the proteins that you need to survive, right? Okay? So instead, as we shall see, a virus relies on hijacking the replication machinery of the host cell in order to propagate itself, right? The job of the virus is to get inside the host cell and use the cell's DNA replication, transcription machinery to make many, many copies of itself, right? At your expense, right? And we'll talk a little bit more about this a little later. Now, bacterial cells can also acquire genes from other bacteria, right? For example, if we mix a laboratory strain of E. coli, which lacks one of the enzymes for making an essential amino acid. Essential amino acid will be an amino acid that the bacterium needs in order to survive, okay? So if you mix two strains, one of them lacks, let's say, essential amino acid number A, see, it'll be non-essential if the bacterium can make it, right? But essential amino acid is something that it will have to make in order to survive, right? So with another strain that lacks one of the enzymes for making another essential amino acid, and if the mixture is allowed to grow together for a few hours, then you transfer it to a medium that lacks both amino acids, okay? So if there was no DNA exchange between the two bacteria, two classes of bacteria, then all of them would die because now both the amino acids are not there, okay? They're missing something essential, okay? But if you do that, you'll see many rapidly growing bacteria can be found in the new medium. And the genome of the new bacterial strain is composed of normal genes for the synthesis of both the essential amino acids, okay? So that means there is some gene transfer that goes on. Yeah? Yeah, a virus is basically an RNA or DNA inside a protein coat. That's what it is. Inside a protein coat, inside a protein cover, okay? There's a protein cover with DNA or RNA inside. That's it. Virus is not a living object, right? It cannot replicate itself, right? It does not have any DNA polymerase or anything. Cell is a living object because it can make more copies of itself, right? It can produce new cell by cell division. Virus is not a living object, right? Okay. Virus is just DNA or RNA. Some of them are DNA viruses, some are RNA viruses encapsulated inside a protein coat. That's what it is. Now, genes can be transferred from one bacterium to another by a process which is called bacterial mating. The ability to initiate mating and gene transfer, seen in some bacteria, is conferred by genes that are contained in what are called bacterial plasmids, right? What are plasmids? Plasmids are small circular double-stranded DNA molecules that are separate from the larger bacterial chromosome, right? And a plasmid that commonly initiates mating in E. coli is what is called the F plasmid or the fertility plasmid. When a bacterium carrying the F plasmid, the donor, encounters a bacterium that lacks the plasmid, a cytoplasmic bridge is formed between the two cells, and the fertility plasmid is replicated, and that DNA gets transferred from the donor through the bridge to the recipient. And finally the bridge breaks down, and I'm going to show that to you in a picture. Finally the bridge breaks down and the two bacteria, both of which now contain the fertility plasmid, can act as donors in subsequent encounters with recipient bacteria. And this is explained in this diagram, right? So here you have these two bacteria. Initially this bridge is not there. This is a donor one because it has the fertility plasmid. This is the bacterial chromosome. The blue stuff is the fertility plasmid. Small DNA molecule or a circular DNA molecule. This has information for making that bridge, right? So this is the donor, that's the recipient. So the information here will be used to produce this cytoplasmic bridge, and then there will be DNA replication, right? See, this thing opens up and it will go in here, and you have replication starting here, right? Then starting here, and at the end you basically have each bacterium has now got a fertility plasmid. Okay, this is called rolling circle replication. This the way in which this thing is replicated, and you get one copy here, another one here, right? It's called rolling circle replication. So you basically first open this out, then you take it here, then you start using that as a template to produce a copy, and then make another copy looking at the inner circle, right? Make another copy. Yeah.
Will the bridge disappear?
Yeah, it will disappear. Yeah. After it's done, then the bridge will disappear. Yeah. Because the proteins that are responsible for producing the bridge, maybe the transcription of that is turned off. Okay, transcription and translation. Yeah, it will disappear. It will not be a permanent thing. Right. Now the problem is with this, you will not have that much of genetic variation because this is a small stretch of DNA. Right. So it's only a limited number of genes that will be transferred from the donor to the recipient. Right. However, there can be situations where this thing actually combines with the bacterial chromosome, right? And then when it leaves the bacterial chromosome, it can take some of that with it, okay? So producing genetic diversity or genetic variation. Now the fertility plasmid or the F plasmid is necessary for mating because it carries genes that encode some of the proteins that are required to make the cytoplasmic bridge and to transfer the DNA, right? And as I said, the bacterial mating via the fertility plasmid does not generate much genetic variation since the fertility plasmid that is transferred contains only a small number of genes. However, occasionally the fertility plasmid can get integrated into the bacterial chromosome, right? With the result that when it now initiates mating and gene transfer, it can now take parts of the bacterial chromosome with it, okay? Because it merges with the bacterial chromosome, but when it excises itself, it will take some part of the bacterial chromosome with it. Also bacteria, so here we are basically talking about all the different mechanisms by which, the purity of the DNA can be altered, It's not going to remain the purebred DNA, right? Because of all these different things that happen. Now bacteria can take up DNA from their surroundings. For example, DNA can be taken up from other dead bacteria in the surroundings. This process is called transformation since it can transform one strain of bacteria into another. And recall that experiment from chapter 5 when you had the smooth strain and the rough strain, right? The rough strain was non-pathogenic, that got transformed to the smooth strain when it was grown in the presence of heat-killed smooth strain, right? So this can happen, okay, where one strain of bacteria gets transformed into another. Now the most important route by which DNA becomes incorporated into a bacterial genome is what is called homologous recombination. Homologous recombination can take place between two DNA molecules of similar nucleotide sequence. They are not identical, very similar, right? A lot of matching up is there. They come next to each other, then they cross over, right? So this is shown in the next figure. So let me go to the figure first, right? So here you have like two DNA strands, double-stranded DNA, right? They come next to each other, very similar sequence, right? Then they just cross over, right? This gets attached to the one at the bottom, and that one gets the green gets attached to the red, and vice versa, right? This kind of crossover can take place, right? And this is referred to as homologous recombination, right? So this is shown in the figure, and the same figure also shows how they can then cross over. The net result is homologous recombination. Cells utilize specialized proteins to facilitate homologous recombination, And such recombination enzymes are well characterized in bacteria, but are only beginning to be understood in eukaryotic cells. Right? But again, I mean, this is 10 years old, so maybe now somebody else might have gotten a Nobel Prize for understanding the details of this. Okay? It's just that I don't know because I don't keep track of all that. Okay, now if you do have these kind of exchanges, homologous exchanges, right? You can have a situation, there is some DNA fragment, this is homologous to the red one here, and then you have two exchanges, all right? And then this green is transferred basically, with two crossovers, the green can be transferred onto the red one. So now you have a recombinant bacterial genome, all right? It's been altered, okay? I mean, these two are similar, but they're not exactly the same, so there is genetic variation here. Another example could be how does the fertility plasmid integrate with the bacterial chromosome? Well, you just need one crossover, because if you get one crossover, this could link up to that side, this could link up here, then you unwind it, right, or you uncoil it, and now you have a bacterial chromosome that includes the integrated fertility plasmid, right? Now later on when this plasmid leaves the bacterial chromosome, right, it might take a little bit of this DNA within, and then, you know, when it when it interacts with another donor bacterium, another recipient bacterium, it will transfer some of the genes from this bacterial chromosome over there. Yeah. No, it happens in eukaryotes also. Okay, I'll talk about that. Yeah, even in your cells it will happen. Okay, when you get a cold, okay, the virus will come and change some of your DNA, right? No escape. So. Now, gene transfer can also be carried out by bacterial viruses. You know, viruses don't just attack you and me; they also attack bacteria, right? So, bacterial viruses or bacteriophages, these are viruses which invade bacterial cells. Now, getting back to your question, who asked me about the virus? Okay, yeah. So, virus is usually composed of DNA that is enclosed inside a protein coat, right? Inside a coat of protein. It could also be RNA. A virus will enter a bacterial cell, and that's true even for our cells also. It will get into the cell and it will use the cell's DNA replication, transcription and translational machinery to produce, number one, more copies of its own DNA and more copies of its coat protein. Okay, so then there is more material to package and make more viruses. Okay. So the replicated DNA is packaged into additional protein coats to produce progeny viruses which can leave the current bacterial cell and invade other cells. Right? And viral reproduction is usually lethal or deadly for the infected cell, and the cell bursts open, lyses as a result of the infection. Like when you get a cold, okay, that runny nose and all that stuff, that is cytoplasm from the cells, right? Because the virus has killed a lot of the cells, okay, made more copies of itself, and you know, left the dead cells behind. Okay. Now, when a virus invades a bacterial cell, one of two things can happen. First, you know, it could be that the cell bursts open and many copies of the virus are released. Okay, that's why they talk about all these diseases. You know, you're going to give the flu to somebody else and all that stuff. You know, because many copies of the virus are released, so somebody else will pick it up, and that's one possibility. The other one is that the virus can sit dormant. The viral genome, the genome of the virus, because remember, the virus contains DNA or RNA. That can get integrated into the bacterial chromosome, and every time the bacterium divides, right, the viral genome is also replicated along with the bacterial genome and passed on, right? Now, at some point in time, you go, I mean, it's lying there dormant, right? But at some point in time, let's say you give an insult, such as exposure to ultraviolet light, could induce the viral genome to leave the host chromosome and begin a lytic phase of viral replication, right? That means kill the cell and release a lot of copies of the virus, right? Now, most of the time it's route 1 that is followed. You know, it doesn't stay dormant. Most of the time it will go there, kill a lot of cells, make more copies of itself, right, and then get out. But sometimes route 2 also is followed. Now, the viral genome usually integrates with the bacterial genome by what is called site-specific recombination, which is carried out by an enzyme that is called integrase. And again, these are just details for your information. You know, it's not, there's nothing really much to understand over there. Now, on leaving a host chromosome, the viral DNA will occasionally remove itself inaccurately and bring along a neighboring piece of host DNA in place of part of its own DNA. Right? And this host DNA will be packaged into a virus particle along with the viral DNA. So the next time when this virus infects a new host, it will introduce along with its own viral DNA that it had picked up from the previous host, right? We are not saying how exactly it will happen or that something systematic has to happen. We are just saying that there are a lot of things that will cause changes in the DNA, okay, over evolutionary time. So this bacterial DNA can now become part of the new host chromosome in at least a couple of ways. Number one, the incoming virus could integrate into the new host chromosome, right? Or if the incoming virus does not destroy the host, the passenger bacterial DNA could become a permanent part of the host genome by homologous recombination, right? And this process is called transduction. So a lot of things, a lot of different, possibilities for messing up the pureness of the DNA, okay? Whether it is in virus or even if it's in us, right? Now, many bacterial and eukaryotic genomes, they contain stretches of DNA that are called transposable elements. They can move around, right? Which can move from place to place within the chromosome by a process that is called transposition. And somebody brought up this topic, I think, last time, whether things can move around. They can, right? So transposons move within the DNA of their host by means of special recombination enzymes, which are called transposons, encoded by the transposable element to create great genetic diversity. And you can have two types of transposition, all right? You could have, like this is the donor DNA, so this is the target DNA. So if it is non-replicative transposition, this thing will be cut out and just put in over there, okay? So this is the new DNA sequence, all right? Target DNA, from the donor you cut this out. In replicative transposition, the donor DNA doesn't change. You just make a copy of this and stick it in there, right? New DNA. Now we know that most alterations in the genome are harmful to the individual bacterium, and these are quickly eliminated from the population, right? If some mutation in the DNA, right, makes the bacterium not viable, right, it cannot survive, then they are gone. However, these alterations also will confer survivability in a changing environment, as was demonstrated by the example involving that antibiotic rifampicin. Right? Now, so we have talked a lot about bacteria, okay, bacteriophages, bacterial transposition and so on, okay. What about the case of eukaryotic cells, okay, what could cause genetic variation? Unlike bacterial DNA, eukaryotic DNA has got both coding and non-coding regions. What are they called? Anybody remember? Okay, good. Okay, so you're getting prepared, right? Or maybe you already knew it from before, okay. So in prokaryotes, the rate of cell division is very high, and hence there is strong selective pressure to minimize the amount of superfluous DNA in the genome, right? So they think that this is the reason why prokaryotic genomes have gotten rid of all that intron DNA, okay? You don't have that anymore. Now, eukaryotic genomes are also characterized by a large amount of gene duplication that has occurred over evolutionary time. As a result, there could be several genes belonging to the same family. And the most well-documented example is the beta-globin gene family. That's one of the subunits of hemoglobin, right? And the human genome has a total of 5 beta-globin genes, right? So these genes encode the beta subunits of the various hemoglobins produced at different times during embryonic, fetal, and adult life. So you have different hemoglobins that you're using, right? When you're at the embryonic stage, when you're a fetus, okay, when you're a kid, and then when you're an adult, okay, all that information is there in your genome. Right? So how does gene duplication occur, right? So they think that gene duplication occurs like this, all right? So if you're going to have homologous recombination, all right, the two genes, this is double-stranded DNA, this is double-stranded DNA over here, okay? With similar sequences, they'll align, okay, and then there will be a crossover. So that causes genetic variation, right? That brings in some diversity. But you might have a situation where the alignment is wrong, okay? Instead of aligning up like this, they align this way, okay? Maybe this sequence and this sequence match up, all right? Now if there is a crossover, then one of these chromosomes, right, will have 2 copies of this gene, okay, from here and from here. The other guy has nothing, right? So the individuals that inherit this one, right, that will not be a functional gene. They are eliminated from the population, right? Nature has its way of, getting rid of non-performers. Okay, if you cannot produce the protein you need, you're gone, right? But the ones that survive, right, they are going to have 2 copies of the globin gene, right? Yeah, go ahead.
So how do you detect the misalignment?
No, I don't detect misalignment, okay? I mean, like, over time it has happened, right? Because that's how they think that it has happened. Because otherwise, why is it that we have 5 different beta globin genes? Okay. I mean, you really needed one, right? I mean, you have 5 different, and probably there are others also. You have many copies of those, right? And this is something that we are not detecting because it has happened over evolutionary time. Okay, thousands, maybe millions of years. Yeah.
But these 5 different genes, they produce exactly the same protein?
Not exactly the same. Little bit different. Yeah. And they are especially well suited for the particular time in your life. Okay. Like if it's a fetus, they're using one. Okay. So there have been some adaptations that have gone on. Okay. So I don't know the answers to those, but if you look at the human genome, you'll find 5 copies of that. Okay. And furthermore, they have done experiments to see that each copy is suited for a particular stage in development. Because these things, like, see, we know about them for very few years. Okay. But like I have a colleague in biology. He says when you're trying to fight cancer, right? These are cancer drugs. These are only 50, 60 years old, or maybe 40 years old. Okay. But he says the cancer cell is very intelligent. Okay, because it has gotten billions of years to learn and fool you. You think you're smart. That one has got so much of history, right? So it's the same thing here. How this developed over evolutionary time, we don't know. But these are just some of the theories. Okay. Yeah, I wouldn't be able to tell because I would not be even able to see that crossover in real time. Okay, because this may happen, it may not happen. Today, with the techniques that we have, the laboratory techniques, all right, we can actually do a lot of these things in the lab. Okay, we don't have to wait for a million years for things to happen. Okay, we can cut out DNA, put it somewhere else, and try all kinds of experiments. Okay, so what I showed you in this picture, gene duplication is thought to occur from a rare recombination event between 2 homologous chromosomes. Consider the case of the beta globin gene duplication. Instead of aligning properly for a crossover, the 2 homologous chromosomes align in an improper fashion as shown in the figure that I showed you. After the crossover, the long chromosome has got 2 copies of the globin gene, while the short chromosome lacks the original globin gene. Consequently, the individuals that will inherit the short chromosome, they'd be expected to be eliminated from the population. While the individuals that inherit the long chromosome, they'd be expected to have 2 beta globin genes instead of 1. Right? And genes encoding new proteins can also be created by the recombination of exons. Right? And the general scheme is the same here, except that it's not just the entire gene, but it's just the coding region that is involved. And I have a slide showing that too. Right? So the same kind of thing can happen involving only exons. Right? This is duplication of exons. So you have exon A, exon B. Right? In between there is this non-coding region, right? So again, I mean, if there was a proper crossover, this exon A would align up with exon A, exon B with exon B. If there's, unequal or improper crossing over, let's say the crossover took place here. Now you have exon A, exon B, right? On the long chromosome, and the short guy only has exon A, right? So you now have exon duplication, okay, not just gene duplication. Now one more thing, note, because you have introns, there is a lot more possibility for crossovers, okay, which will not mess up the coding regions, right? So the presence of introns in eukaryotic DNA greatly increases the chances of this kind of recombination. Because if stuff gets messed up, if you cannot produce the correct protein and all that, then that that particular cell will be eliminated from the population. That organism will be eliminated. But because you have introns, that makes it possible that you'll have crossovers and you will still be functioning, right? You still have functional genes. So the general scheme for gene duplication, which is shown in the figure that I showed you 2 slides later, is the same as before, except that an exon within a gene rather than the entire gene is duplicated. So, and this is just stating what I mentioned. Without introns, there would be very few sites on the original gene at which a recombinational exchange between homologous chromosomes could duplicate the domain without damaging it. Therefore, introns greatly increase the probability that DNA duplications will give rise to functional genes encoding functional proteins. Right? And moreover, the presence of introns greatly increases the probability that a chance recombination event can generate a functional hybrid gene by joining together 2 initially separate exons coding for quite different protein domains. And this is referred to as exon shuffling. Right? Exon A, exon B, exon C from somewhere else, you combine them together and still it will be functional. So the introns make those exchanges possible. Now about 10% of the human genome consists of 2 families of transposable sequences. Now transposable DNA elements, they move from place to place by the mechanisms that have been discussed earlier for prokaryotic transposons. All right. Remember we talked about replicative transposition and non-replicative. Same kind of thing happens here also. However, for eukaryotes, there are also what are called retrotransposons, for which an RNA copy is first made using RNA polymerase, following which DNA copies are made using reverse transcriptase. Right? Why is that? Right? So let me show you in the picture. Right? So you have the donor DNA, right? So that is going to get copied somewhere, right? But see, this will have both coding and non-coding, right? So before doing that, if you use RNA polymerase from the DNA, it can transcribe the DNA, give you RNA, right? But in the case of eukaryotes, that RNA will become a messenger RNA or a continuous coding sequence. Then you use reverse transcription to get the complementary DNA, right? So you have gotten rid of all the introns, right? And now this can be used and inserted into target DNA, right? So there are also retro transposons, okay, that will first make an RNA copy, then go back, and the intent here is basically to get rid of the non-coding region, right? So for eukaryotes, there are also retrotransposons for which an RNA copy is first made using RNA polymerase, following which DNA copies are made using reverse transcriptase, and it is these DNA copies that are then inserted into the target. Now transposition by reverse transcription is shown in the figure that I pointed out a short while ago. And examples of human retrotransposons are the so-called L1 transposable element and the ALU sequence. Again, for us, these are just some names, okay? I mean, I don't know the details about these individual retrotransposons. Now, the evolution of genomes has been greatly accelerated by transposable elements, okay? Because they're causing changes, you know, like you put in new DNA somewhere, you might have messed something up. You might have messed up transcription, okay, or translation or whatever. So the insertion of a transposable element in a regulatory region for a gene will often, by disrupting or adding short regulatory sequences, have a dramatic effect on gene expression. Now, another source of genetic variation in eukaryotes is the activity of viruses. Just like in bacteria, you have bacteriophages that can come in the case of eukaryotes also. And we know that we catch cold all the time, right? I mean, flu, cold, and all that stuff, you know. So like bacteriophages, viruses that infect eukaryotic cells are fully mobile elements that can move into or out of our cells. And viral genomes can be made either of DNA or RNA and can be single-stranded or double-stranded. An important class of viruses is what are called retroviruses, which reverse the normal flow of genetic information. The normal flow of genetic information is DNA to RNA protein, right? If you look at the HIV virus which causes AIDS, that's a retrovirus, okay? It has got RNA, so RNA inside a protein code. So that RNA has to be first converted into a DNA by reverse transcription, right? And then it's going to integrate into the genome of the host cell. So the retroviruses have a genome that is made of RNA and have a protein code that encapsulates the genome. And also this enzyme reverse transcriptase, because from the RNA you have to make the DNA. You need that enzyme too, right? So the schematic diagram for a retrovirus hijacking a host cell is shown in the next figure, right? So here you have a retrovirus. So this is like the RNA, the blue one is the RNA inside. Then you have the reverse transcriptase because that's needed to convert from RNA to complementary DNA. Then you have this protein coat, and there'll be some lipids and all that in the boundary of this. So the way it's going to attack the eukaryotic cell is that the boundaries will fuse, okay? So this thing will come out, then this guy will get in, okay? Now, then from here you have the RNA, then you have the reverse transcriptase. From RNA, so RNA and will be acted upon by reverse transcriptase, right? Reverse transcription to give you complementary DNA. So it will give you single-stranded DNA, right? That's let's say the red one. Then, so the blue one is the RNA, okay? The RNA can still be degraded, but this complementary DNA that you have, that's a much more stable molecule. That can be worked upon by DNA polymerase, which is there in the cell, all right? And it'll make it double-stranded DNA, viral DNA. Right? Now, so from that RNA, you now have double-stranded viral DNA. That can get integrated into the host chromosome, right? Every time the cell divides, right, it'll make copies of this too, okay? Now, when this eukaryotic cell does transcription and translation, it will produce that RNA, right? It will produce that RNA. When it does the translation, it will produce many other proteins, but it will also produce these viral proteins, these green guys, all right? And so what is going to happen when this cell is killed, okay, when the next generation of viruses is released, there will be many more of these that will be formed, just like this one, right? and in the process the cell got killed. Okay, that's what happens when you get a cold. Okay, virus. Yeah.
The viral DNA only produce this viral copy of itself, or it does something else. I mean,
No, the coat will come from here. Yes, there is enough information in there for producing that protein coat. Okay, it will have that information for producing those proteins. Okay, by
at the end we only have this virus which is copied, and we have the copies of the virus, or there is next generation of it. I mean,
No, it will be many copies of the virus. Yeah, copies of the virus.
the
Yeah.
same, right?
Yeah, but they might be a little bit different. Like, I mean, it might, when it came out, maybe there is something else that got attached or something. There could be some variation, but in general, it will be many. The goal is here is to produce many copies of this virus. Yes.
Virus, the RNA already have transcription.
Why? Because you need to produce DNA, reverse transcriptase, all right?
I mean, he already
Oh, why the bacteria already has RNA polymerase? Is that what you're saying? Or DNA polymerase?
can he produce DNA in a virus cell?
No, because they don't have the replication machinery. The virus, it just has this, okay? It does not have the ability to make more copies of the RNA or DNA or anything, you know. That's why it needs your cell, okay? That's why it needs your body and to make you sick and make more copies of it. And if it could do it by itself, then it doesn't need to infect you. So in the case of retroviruses, the enzyme reverse transcriptase is first used to make a single-stranded complementary. This is what I showed in the picture. Complementary DNA copy of the viral genome. Then the enzyme DNA polymerase present in the host cell is then used to create a double-stranded DNA copy of the viral genome, which is then integrated into the host DNA. Then transcription and translation from the integrated genome produces copies of the viral RNA, reverse transcriptase, and the coat protein. And all of these can be packaged together to produce additional retroviruses. Now, retroviruses that have picked up host genes can make cells cancerous, right? Very few human cancers are caused by retrovirus infection, but these viruses are a prominent cause of cancers in some animals. And the best example of that is what is called the Rouse sarcoma virus. Right? Sarcoma is a tumor. It's a big tumor, right? Big cancerous tumor, right? So this virus can pick up a gene from its chicken host. Okay, it infects chickens. This gene, the gene that it picks up is called SRC. It is unnecessary baggage from the point of view of the virus, but it has got profound consequences for cells that are infected by the virus. The normal SRC gene in the chicken genome, it encodes a protein kinase. What does a kinase do? Does anybody remember?
polymerase.
Uh-uh.
It kills proteins? No.
No. You need to study, okay, all of you. It adds a phosphate, yeah. So this guy is studying, okay. So associate with him, okay. I'll study more. Anyway, so it adds a phosphate group, and as I said before, you know, eukaryotic cells have the property that by adding a phosphate, right, change the shape of the protein a little bit, okay, and turn it on, okay, or off, okay. And in fact, that is how eukaryotic cell division is controlled. So this is a protein kinase, this normal SRC gene. So it will turn on cell division in response to the right stimulus, okay. But oftentimes, so it is called a proto-oncogene because it's a gene that hasn't become cancerous, right? But so it responds to the right stimulus to turn on cell division. But the SRC gene that is carried by the virus, when it picks it up from the chicken host, it might have messed it up a little bit. It's not quite identical to the normal cellular gene, and it may be turning on cell division, right? When it's put back in the chicken, even when no new cells are needed, okay? And this difference gives rise to its ability to cause a cancer. The mutated SRC gene is called an oncogene. Right? Oncogene is a cancer-causing gene. Right? It will turn on cell division even when there is no stimulus. Right? And it causes the infected cell to divide uncontrollably. So this is an example of a cancer that can be caused by a virus. Right? So very few human cancers are caused by viruses. But in case of animals, there are examples. Okay, now we get to the issue about your question about why, you know, the two whatever siblings are not going to be identical. Okay. So now bacteria reproduce asexually, and this gives rise to offspring that are identical to the parent because it's just a fission type of cell division, right? Sexual reproduction, on the other hand, involves the mixing of genomes from two individuals to produce offspring that are genetically distinct from one another and from both their parents. Sexual reproduction occurs in diploid organisms. You have to have 2 copies of each chromosome, in which each cell contains 2 sets of chromosomes, one inherited from each parent. The actual cells that carry out sexual reproduction in diploid organisms are called the germ cells or the gametes. Right? These gametes, which are haploid cells, are of 2 types, the egg and the sperm in animals. So, the egg and the sperm will have 1 copy of each chromosome. Right? All other cells, they have 2 copies. Okay? So the particular copy that is passed on to the egg or the sperm, that's at random. Okay? So the haploid germ cells are generated from the diploid precursors by a special type of cell division which is called meiosis. Right? And we'll talk about meiosis and mitosis later on. But here I just want to show you what the odds are of 2 people having the same genome. All right? So our main focus here is on showing the staggering amount of genetic variation that results from meiosis. A schematic diagram showing how meiosis produces haploid gametes from a diploid precursor is shown in the figure. So let's go to the figure. A figure is worth 10,000 words, not just 1,000. So here, let's say this is a cell, all right? This is a germ cell precursor, right? And we are focusing only on 1 pair of chromosomes, all right? Paternal chromosome, maternal chromosome. There are 23 pairs like this, all right? Now, meiosis, it involves halving of the number of chromosomes. So if from this cell, if you have to produce the egg or the sperm, all right, the gamete, all you needed to do is basically split the cell in 2 and get one of these to go into one cell and the other one into the other, okay? For some unknown reason, that's not how it happens, all right? Basically, there is DNA replication and then there will be 2 rounds of cell division, okay? That's the way it happens. So you have DNA replication, the paternal one is replicated, the maternal one is replicated. Not only that, after that, paternal and maternal chromosomes, they are homologous chromosomes, right? Like chromosome number 1, paternal, maternal copy, they're homologous. They will line up next to each other, right? And there are crossovers also that will take place. So there is more genetic variation over there, okay? And then once, the part of this one has gone, right, see? This portion has now gone to the bottom one, okay? Now there will be 2 rounds of cell splitting, okay, to produce 4 of these, 1, 2, 3, 4, right? So there will be 4 different types of gametes, okay, that will be produced. So there are 2 rounds of cell division, 1 division here, then another round, so you have 4 different types of possible gametes, okay? This is for 1 chromosome, right? There are 23 pairs of chromosomes, right? Even if you didn't have any crossovers, so if you have crossovers, you're getting 4. If I didn't have any crossovers, I would have 2 different types of gametes, okay? So on 23 chromosomes, if you look at that, the total number of possibilities is 2 to the power of 23. On an average, there is at least 1 crossover on each chromosome, right? Now the egg and the sperm will fuse. So there are 2 to the power of 23 possibilities for eggs, 2 to the power of 23 possibilities for sperm. Okay, you fuse them together, look at the number of possibilities. And that is without crossovers. If there are crossovers on an average, it'll be like 4 to the power of 23, okay? So the odds that, 2 people will get the, unless it's an identical twin, you get the same genome are almost 0, so.
The other 22 chromosomes are the same, right?
What?
The other 22 chromosomes, they are the same.
No.
No?
22 chromosomes.
I mean the other 22 pairs of chromosomes.
No, the same thing will happen with.
Each pair are the same, right?
No, it's just like this one. All of them, it's like that, okay? Okay, let me explain, then we discuss, So, yeah, go ahead.
You have 4 different
No. Yeah, four different.
The other 22.
See, from this one you got 4, right? From 1 chromosome, right? 1 chromosome pair. Each chromosome pair you'll have 4 different possibilities, okay? Including crossover. Each chromosome pair. So if it is 23 times, it will be 4 to the power of 23. Okay, different possibilities. Like on chromosome 1, okay, you got the paternal, maternal, okay, after the crossover there are 4 possibilities, so in this location. In the chromosome 2 there are another 4 possibilities, okay, and this is just the egg or the sperm. Another one, 4 possibilities. If you don't allow crossovers, every chromosome there are 2 possibilities, okay, of what you're going to inherit, right? So the numbers are really huge. And we will see this again a little bit later, because I will talk about cell division also, right? So mitosis and meiosis. So we focus attention on only 1 pair of homologous chromosomes, like I did in that picture. During meiosis, the chromosomes first duplicate, and then the homologous paternal and maternal chromosomes line up next to each other. Next, crossovers take place by homologous recombination, and 2 rounds of cell splitting occur to produce 4 different haploid gametes, okay, which is what I showed in that picture. So clearly, in the absence of a crossover, each pair of homologous chromosomes will give rise to 2 different types of gametes. Okay? If I didn't have any crossover, instead of 4, I would have 2, depending on whether it contains the paternal homologue or the maternal homologue. With 23 chromosome pairs in the human genome, each individual can produce 2 to the power of 23 possible gametes. The actual number of possible gametes is much higher because of crossovers, and on an average, there is at least 1 crossover on each chromosome. So to develop a new organism, 2 such gametes of the opposite kind, egg and sperm, have to fuse to form the single diploid cell that will undergo repeated mitosis, right, repeated cell division to develop into the new organism. Thus, the number of possible ways in which the genetic material gets reshuffled during sexual reproduction is staggering. And that is the reason that it is not at all surprising that no 2 individuals except identical twins have the same genome. Hopefully I answered your question at the beginning of the class, okay? So now you don't have to think about, it's just not going to happen, okay, that 2 people are going to have the same genome. Very, 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 very unlikely. All right, any other questions? So I'm done for today, right? So next time we will talk about DNA technology, because this is variation that takes place in nature, okay? But in this day and age, especially in the last 40 years, people have developed techniques where they can go and do all kinds of things. You can cut out DNA from one organism, put it in another, right? You've already seen that, growing eyes in the middle of the legs and things like that, So, and that is what is going to be important for, and that's where the double E's and all will come in, okay? Because taking the data from here and analyzing that, and there's a lot of demand for, people with quantitative skills, okay?